So, hello everyone. These are our second discussion. So, if you are new in our channel, please subscribe our channel, and we will upload all the previous year question explanation. And you know that previous year question explanation will help you for boost your preparation also. And many questions previous year questions are almost repeated this year. Also, some of the questions are repeated, so please and also if you are preparing for gate exam, we are also uploading some gate related questions or gate related previous year explanation. So please subscribe our channel, and so that you get more updated in that if you are preparing for CSI net, right? So let's start our second discussion. So first is the, see that question is the following statement and were made to describe the role of the free energy. We know that at delta G in the represent delta G equal to del H minus T delta S, right? Delta G equal to del H minus T delta S, and reaction will be spontaneous if the delta G equal to negative. Now delta G equal to negative, then reaction will be spontaneous. So reaction can take place spontaneously if the del G is negative. This is correct. Reaction can take place del G is positive, so B is wrong. So you can eliminate B from here. Now you can see that C. Now C and D. Delta G provide no information about the rate of a reaction, and as delta G provide no information about the rate of the reaction. These are correct option. So a negative del G indicates that a reaction can occur spontaneously, but it does not signify the whether it will proceed at a percentable rate, right? So that is the answer will be, so A and C is the correct answer. So del G provides no information about the reaction of the rate of the reaction. So A and C is the correct answer. Now, this question, for an exponential growth culture of bacterium in zero in the initial population number, nt is the population number at time t, the mean growth rate k is expressed. We know that formula n equal to n0 into 2 to the power n equal to n0 into 2 to the power n. So n is the number of at the end of the time interval. So n is the number of bacteria at the end of the time interval. Now n0 is the number of bacteria at the beginning of a time interval, that is initial number of initial number of bacteria. And n, small n, is the number of number of generation number of generation, that is number of times of the cell population doubles during the time interval, right? So we know that n equal to n0 into 2 to the power n. Now we are applying both side log, so log n equal to log n0 plus n log 2. Now log n equal to log n equal to log n0 plus n log 2. Now from here, the number of generation n will be number of generation n equal to log n minus log n0 by log 2. Now here is the, now you have to calculate the growth rate constant, mean growth rate constant. Now growth rate constant k is the number of generation per unit time. Now log 2 value is, so k will be k equal to n by t actually, right? In case of here, you can see that, so n, it will come log n minus log n 0 by log 2. Log 2 value is 0.301. Now 301, now you have to this one, this is the number part time, so you have to also multiply log n minus log n 0 by 0.301 into t. So this will be the answer. This will denoted as k. These are the mean growth rate constant k. So this option 1 is the correct answer. Log n t minus log n 0 by 0.31 t, right? Now next question. So in C elegans we have seen that question said apoptosis pathway. This will be EGL1 inhibits Z9, Z9 inhibits Z4, Z4, Z3 and apoptosis, right? This option will be correct answer. Now we'll see the next question. Now, in case of CDK activating kinase, this will be the option 4. Phosphorylates an active site in the CDKs or cyclin dependent kinase. V1, V1 actually, in case of V1 kinase, it's a phosphorylates inhibitory sites in the CDKs primarily involved in suppressing CDKs1 activity before mitosis. Now, P27 suppress the G1A CDK and A CDK activation in G1 helps cell withdraw from cell cycle when they are terminally differentiated, and phosphorylation by CDK2 triggers its ubiquitination by SCF, right? And P21 is a suppressor of G1S CDK and S CDK activities following DNA damage. So option 1 is the correct answer. Now see the next question. V snare and T snare. Now V snare for vesicle, T for teether, right? V snare and T snare. In case of hair, you can see that now V snare transmembrane protein exit as complementary sets with V snare on vesicle membrane and T snare on the target membrane, right? These are correct. V snare is the vesicle membrane and T snare in the target membrane. Now you can see V snare is usually composed of, no, these are three protein. T snare is not possible, not. Then V snare T snare protein of a pair interact via helical domain proposed by the two protein, resulting in the formation of the stable double helix. This also not correct. So membrane fusion is catalyzed by the energy that is freed when the interacting helix wrap around each other to pull the membrane faces together and concurrently squeezing out water molecule from the interface. These are the correct. So A and D is the correct option. Now see the next question. The following statement about are being made about the archaeal bacterial cell membrane. We know that archaebacteria is the primitive type of archaebacteria is more closer to eukaryotes, right? And so first see that archaeal cell wall contain, basically archaeal cell wall contain and could stain gram positive and gram negative depending on the genus. Okay, these are correct depending on the genus. Now archaea are characterized by gram positive staining on the cell. Archaeal cell wall susceptible to degrade by lysozyme. Archaeal cell membrane possesses branch chain hydrocarbon linked to glycerol by ether link. Yes, these are ether link present. In case of archaea have ether linkage. Archaea have ether linkage, ether linkage, branch aliphatic chain, branch aliphatic chain in their membrane lipid, branched aliphatic chain in their membrane lipid. membrane lipid. Right now, archaeal cell membrane possesses branch chain hydrocarbons linked to glycerol by ethyl links. Right, these are correct. So A and D is the correct answer. Right. So this and in case of normal, you in bacterial have ester linkage. In case of bacteria have in their ester linkage in their fatty acyl chain. Right, ester linkage present. Ester linkage. Now, see the next question. Now, in case of here, now RNA pol 1, we know that at pol 1, the RNA polymerase 1 synthesize rRNA. Most of the rRNA are synthesized by pol 1, pol 2 synthesize mRNA, and pol 3 synthesize tRNA. These are the some this is basic, but at pol 1 here, pol 1's transcribe now here is to 85 45s and 18s rRNA, right? Now pol 1 actually now see that second miRNA generated miRNA genes are transcribed by pol 2 RNA polymerase 2 microRNA synthesized transcribed by RNA polymerase 2. The polymer RNA polymerase found in plants, basically exclusively found in plants is RNA pol 5. Pa- and then most of the RNA pol 5 is the pol 4 also present, but most pol 5 and pol 4 both are present, but in case of here these are RNA pol 2 is the miRNA generated synthesized transcribed by RNA polymerase 2. And tacrNA, what is tacrNA? TacrNA is trans-acting siRNA, abbreviated as tacrNA, are class of small interfering RNA, that is siRNA, that repress gene expression through the post-transcriptional gene silencing in land plants. Um, so answer is option 3. Now see that in case of pathogenesis, related, the pathogens continuously evolve strategy to evade host immune response. For each of the following evasion strategy listed in the column, match the pathogen and the which adapt is changing the antigen expressed on their surface. And in here is the, so look at that first one. So in changing the surface, basically the viruses, those are virus, basically they can change their antigen presentation in the on their surface, right? Increasing phagocytic activity of macrophages. Increasing phagocytic activity of macrophages is the gram positive bacteria, is the 3. And C is that developing resistant to complement mediated lysis. This will be in case of B. Here you can see that B will be increasing phagocytic activity by macrophages, no bacteria. This will be no bacteria. And C will be the gram positive bacteria developing resistant to complement mediated lysis, gram positive bacteria. D. Secreting protease to inactivate antibodies, Neisseria. And E is the one allowing point mutation in the surface epitope resulting in antigenic drift, is the influenza virus, right? So answer is option 2 is the correct answer. Now next question. So in case of engrel, the continued expression of engrel and wingless is maintained by interaction between the engrel and wingless expressing cell. The following statement are given towards the initiation of cascade of events that occurs for this interaction. Now, engrel gene is expressed in cells where neither even skip or Fushitaraju gene is active. Now, you can see that engrel will be expressed, we have discussed in this question and previously, so you can watch that video also. So you can see that engrel will be expressed when where even skip and Fusitaraju is expressed. Even skip and Fusitaraju, higher expression of even skip and Fusitaraju in green will be expressed. And lower expression of Fusitaraju there, low or no expression of Fusitaraju or even skip there, wingless will be expressed. So see that question. Engril gene is expressed in cell where neither even skip nor Fusitaraju is active. So this is wrong, absolutely wrong. So you can see that. So option A is gone. These are wrong. So A gone from here. Now B, you can see that the wingless gene is expressed in those cells that contain high concentration of either even skip or Fusitaraju. No, even wingless is expressed where there is no expression of even skip and Fusitaraju, right? So B also gone. So only option C and E is present, right? So C, you can see that wingless is a secretory protein and diffuse to the surrounding and binds with the Frizzle receptor and LRP6 receptor proteins and activate engril via armadillo. Yes, these are correct. Hedgehog protein activate transcription of engril and also activates its own transcription. And hedgehog protein, basically engril does that. Engril actually synthesize, and engril have a auto regulation, right? Engril activates own transcription, and not hedgehog here, right? So a hedgehog protein diffuses from the cell, binds to the patch receptor on neighboring cell, enable the transcription of wingless gene. Yes, these are correct. So you can see that here. These are, so first is the event skip and Fusitaraju, where engril will be expressed, and wingless will be expressed where there is no Fusitaraju and no event skip is there. Another is that you can see that the patch receptors here. So frizzle receptor wingless binds into the frizzle receptor. So these are GSK3 signaling armadillo beta catenin. So engril have own regulation. Engril transcribe and auto regulation is there. So synthesize hedgehog protein. Hedgehog binds into the patch receptor. Cubitus interrupts the wingless synthesis, and wingless binds into the frizzle receptor. So answer is C. And these are C and E will be the answer. Last option. This will be the answer, right? So these are answer is C and E. Now this question, you can see that a large patch of forest area was devastated by raging fires. After some years, the area was found to recover its species. Which one of the option is best describe the process of re-establishment in this area? This question have some tricky. Many of you have done many of you can do mistake here. Basically mosses and lichens, because mosses and lichens actually um, moss actually they are actually decomposer. So here decomposer is not required because already these are have a forested area, right? So and there is a raging fires is there. So you can see that this will be first is the grasses, then herbs, shrubs, and woody plants. These are option 4 is the correct option, because there is no requirement of decomposer here. Because soils are already decomposed by other trees, because already there is a forest, right? This is a secondary succession is going on. So if it is a primary succession, then this will be the correct option. Mosses, lichens, grasses, shrubs, herbs, and woody trees, right? So answer is option 4 is the correct answer. Now, in case of a Kelvin-Benson cycle is divided into three phases, mainly carboxylation, reduction, regeneration. The following statement are related to three phases of Kelvin-Benson cycle. Now, three phases are like in case of air, first is the RUBP. We know that in case of Kelvin cycle, RUBP plus 6 CO2, 6 RUBP plus 6 CO2 together form 12 molecules of PGA. These are phase is called carboxylation phase in presence of Rubisco enzyme. Now from PGA it will synthesize 12 PGALD, phosphoglyceraldehyde. Here there is a use of NADPH to NADP. Another is the ADP, ATP to ADP occurs. Here 12 molecule of NADH, 12 molecule of NADH and 12 molecule of ATP become 12 molecule of ADP. 12 PGLD then divide into 10 PGLD and twice PGLD. Twice PGALD synthesized via, these are in case of glycolysis pathway, and synthesize glucose molecules, right? Now here this step, up to this step 10 PGALD, this step is known as reduction step. The first step is carboxylation. These are reduction, and this one 6 RUMP and 6 RUBP. This one is the regeneration phase. Regeneration phase. So first is the here, so it's synthesized glucose molecule, right? So see that question option, the product of the light reaction ATP NADH is utilized in the carboxylation phase. No, these are ATP and NADH is utilized in the reduction phase, not in the carboxylation phase. So option 1 is gone. Now see that B and D. So 6 molecule of 3-glycerate is converted to 6 molecule of glyceraldehyde in 3-phosphoglycerate in the reduction phase. Yes, these are correct. B is correct. And now another is that the formation of 7 carbon compounds pseudoheptulose 7 phosphate takes place in the regeneration phase. This is also correct. The action of allulase enzyme for the production of fructose 1,6 bisphosphate takes place in the reduction phase. No, actually this allulase enzyme works here. Allulase enzyme, what it does, dihydroxy acetone phosphate is the reaction in glycolysis plus glyceraldehyde 3 phosphate. They join together in presence of aldolase enzyme, produce fructose 1,6 bisphosphate. fructose 1,6 bisphosphate fructose 1,6 bisphosphate, then it converted to fructose 6 phosphate, fructose 6 phosphate, then glucose 6 phosphate, then glucose, then glucose. So answer is option B and D is the correct answer. And pseudoheptulose 7 phosphate takes place in the regeneration phase, right? Now see the next question. In E. coli were simultaneously infected with 2 strain of phage lambda. Now here what you have to do is just find the highest number. So highest number is H plus C plus ST plus. These are parental combination. H plus H, C and ST. So these are 350 and these are 300. Now say your DCO product, these are parental combination. DCO double crossover is the lowest number, H plus C plus ST. Another is the HCST, ST plus, right? HCST plus. These are 6 and 4, right? Now how to will find the middle gene? Middle gene, if you have to find the middle gene, so middle gene is the which you have to compare with the parental combination. Now compare with parental combination. H plus C plus ST plus H plus C plus ST. So change is here. So ST is the middle gene. So you got the ST is the middle gene. From that also you can conclude that here the option 2 will be correct. So more confirmation if you need, you can do another cross also. See that. So ST you have got middle gene. So H plus ST plus C plus, and here H ST C. Now just cross H plus St C and H St plus C plus H plus St C. This are value you can see that H plus St C, H plus St C. These are 86, and this one is 86, and this one H S C plus St is the 114, right? So add them. So 200, 200, and now you have to map distance between H and St. Map distance between H and St will be, you can add 200 plus, you have to add DCO product by 1000 হচ্ছে total progeny into 100. So 210 by 1000 into 100, so 21. So you got that, your confirmation. So option B is the correct answer. So it will be 21 map unit or 21 centimorgan, right? So H and St distance. So answer is option 2. Now see the next question. In case of PR protein, it will be A. So PR2 involving beta 1,3 glucanase, and in case of PR5, it will be thiamutine like, and C PR12, it will be defensine, and PR14 D, it will be 3. It's not possible to remember, so but this question, most of the questions this year, most of the questions are from plants. There are some option are related with plants, like in case of plants, important of plants replicating enzyme, most of the important plant replicating enzyme, these are most of the questions are connected with plants, right? So see that option, which option will be correct? A4, A4 B2, A4 B2 C3, A4 B2 C1 D3. So option 3 is the correct answer. Now next question. In case of programmed cell death or PCD, programmed cell death play important role in development of alluron layer in the following with respect to the various involvement of the phytochromes and the signaling molecules. Which of the following is correct? Now, gibberellin is from a paper that gibberellin acid promotes programmed cell death and abscisic acid postpone programmed cell death, and these are actually from a paper, and these are option A and B is the correct answer, right? So these are actually correct option. So today we'll stop here, and next session we'll discuss next of the question, right? So thank you.